Thanks for the kind introductions. So my name is Yongjun Tan. I work in the Zhang lab at Pairwise University. I'm so glad to share my recent research on Liberibacter here. Liberibacter bacteria are known to be associated with several severe plant diseases around the world, including the citrus Huanglongbing and potato zebra trip. Both diseases, there is not efficiency therapy are available now, and not resistant strain for both plants is found. So, because these two bacteria are intracellular bacteria, and they could be only transmitted by the insect raptor. So it makes experimental study very difficult. As a result, the molecular mechanism of the pathogenicity are largely unknown, especially what are the toxins that cause this plant disease. Further, this bacteria, the Liberibacter, have very strict host specificity. For example, some haplotype in Liberibacter solanacearum could infect and cause disease in potato and tomato, while others could infect and cause disease in carrot and celery. However, interestingly, the cross infection could not cause any plant disease. Therefore, we want to know what genes could contribute to this host specificity in Liberibacter. Here, given the functional studies are largely unavailable, we want to answer these two questions by using comparative genomics. To answer this question, we first conduct phylogenetic analysis for the Liberibacter pathogen and their close-related species. We found that all intracellular pathogens could form the monophyletic clade, while the outside group of all pathogens is the bacteria called the Liberibacter crescent. This bacteria serves as the ancestral group of all pathogens. But interestingly, these bacteria are non-pathogenic and free-living bacteria. Based on this pattern, that is, the ancestor is non-pathogenic, but the descendants are pathogenic. We hypothesize that there are some unique genes, including the virulent factor and toxins, which were introduced at the common ancestor of all pathogens. And we believe that this toxin could contribute to the pathogenicity in Liberibacter. To identify such newly introduced genes, we first study the prophage loci in Liberibacter, because the prophage usually carry different virulent factors or toxins for many bacterial pathogens. Here, we extract all prophage loci in Liberibacter, annotated their prophage component, and classify them into 4 major groups based on their gene composition and phylogenetic relationship. By studying their distribution pattern, we found that the LC1 prophage and the UT prophage only found in limited Liberibacter species, while the LC2 prophage are found in all Liberibacter species. However, the SC prophage found in all Liberibacter pathogens, but not in the non-pathogenic species. Based on this pattern, we believe the SC2 prophage may be introduced as the common ancestor of all pathogens, and they may contribute to the pathogenicity in Liberibacter and may cause the plant disease. Next, after we exclude the prophage component, we conduct the Ortholog analysis for the genomic genes. Basically, we want to identify the genes only present in all pathogens, but not in the non-pathogenic species. We believe that this gene are the newly introduced gene as the common ancestor of all pathogens. If the gene only present in the non-pathogenic species but not in the pathogen, we believe that they are lost in all pathogens. Based on that, here we identify over 300 author group genes are lost in all pathogens. Well, 35 genes are gained at the common ancestor of all pathogens. Among these 35 genes, we conduct sequence structure analysis and uncover several potential toxins. For example, we found 3 components to be peptidase inhibitor. One component belonging EP nucleus family and one component belonging YDJN nucleus family. Based on that, we propose these potential toxins may contribute to the pathogenicity in Liberibacter and cause the plant disease. Further, we also want to answer what genes contribute to the host specificity in Liberibacter. Here, we study different haplotype genomics in Liberibacter solanacearum and conduct the phylogenetic analysis. We found that there is not significant difference between different haplotype genomics in terms of presence of the toxin component and the prophage loci. Instead, by using pairwise comparisons for the genomic genes in different haplotypes, we found the majority of the proteins are highly conserved. Well, there are 18 author group genes display high variation. This indicates that they are fast evolving during evolution. By sequence and structure analysis, we found that 40 genes present in the cell membrane, including one adhesion component and one polymorphic toxins that we just identified. We believe that this membrane protein may be responsible for interacting with the host proteins and mediate the host specificity.